Hello, my name is Ashley Malkin. I'm a sophomore at Greenwich High School, and I'm so excited to talk to you today about my project, Identification of Therapeutics for Neurological Disorders through Development of a Novel Machine Learning System for Predicting Drug-Gene Interactions in the Glymphatic System. Sorry about that. Oh, here we go. Okay, there, got the slides working. Here's a quick overview about what I'll be talking with you about today. The goal for my research was to create a machine learning system to find new treatments for neurological disorders, which can have devastating impacts on millions of people around the globe. In doing so, I was targeting a newly discovered biological system, the glymphatic system. My research had three stages. The first stage created GLYMDS, a new glymphatic-specific dataset. The second stage created GlyMRX, a novel machine learning system that employed GLYMDS for drug discovery to find new treatments for neurological disorders such as strokes, Alzheimer's disease, and epilepsy. And a final stage was implementation, where using GLYMDS, GlyMRX identified new real-world drug-gene pairs for potential medical use. The glymphatic system has been recently identified as a likely origin for a wide array of neurological disorders. It was only discovered in 2012, and it functions as the waste clearance system of the brain. You can see in Figure 1 a diagram of how the glymphatic system functions, with an influx of cerebrospinal fluid, abbreviated as CSF, that's carrying waste products, toxins, and other products, and then mixes with interstitial fluid, or ISF, and then flows out of the glymphatic system through neurovascular units or perivascular channels. Because of the glymphatic system's important role in waste removal, it's been linked to a number of neurological disorders, including strokes, Alzheimer's disease, and epilepsy. For example, since Alzheimer's disease has been linked to amyloid beta plaques forming on the brain, the glymphatic system, if functioning properly, is what removes these plaques and keeps them from building up and causing symptoms. Machine learning has emerged as a tool to aid chemists and biologists in the field of drug discovery. Machine learning can process huge amounts of data and find correlations much faster than a human or even team of humans can. Machine learning models are typically trained on large biomedical ontologies and scientific datasets from the NIH and other organizations, and machine learning could be a vital tool in finding new treatments for glymphatic system irregularities. However, since the glymphatic system is so new, what happens when this data doesn't exist yet? Since the glymphatic system is so newly discovered, there are no databases of glymphatic-related genes and drugs. Compare this to another very important area of study, such as cancer. If you're building a machine learning model to find new treatments for cancer, then you can use resources like CancerRxGene, NCI Cancer Genome Atlas, CTR Database, CancerDR, and there's hundreds of identified genetic drug targets that you can use as your training dataset. Compare this to the glymphatic system, which has none of these resources yet. To remedy this problem, I created GLYMDS in the first stage of my research. The first step in doing so was to identify the relevant proteins to include in GLYMDS. Since the glymphatic system is so new, there isn't even a search term or category for the glymphatic system on these major biomedical ontologies. Instead, I individually selected proteins based on their location and function. I used proteins from the glymphatic system in areas like the blood-brain barrier, cerebrospinal fluid, perivascular space, and microglial cells, as well as proteins from waste and toxin clearance systems in other areas of the body, including the lymphatic system, since drugs that work in one body system historically can work in similar body systems, and the lymphatic system and glymphatic system have many similarities, such as that they're both circulatory systems for waste removal, and the glymphatic system even drains into the lymphatic system. I used 4 different databases to create GLYMDS, the new glymphatic dataset. I first identified glymphatic-related genes on gene ontology using the process that I detailed in the slide prior, and then identified what the gene outputs of these genes were, which were proteins, and then found those protein structures on AlphaFold. I identified the drug-gene interactions from these drugs on the drug-gene interaction database, and then got the structures for those drugs on DrugBank. It's important to note that these drugs weren't created with the intention of the glymphatic system in mind, but since they interact with these glymphatic-related proteins, they're a good starting basis to find other FDA-approved drugs that may also interact with the glymphatic system. All of this together creates GLYMDS, the new glymphatic dataset. And using the new glymphatic And using the new GLIMDS dataset, I developed GlimmerX, a novel machine learning system that uses 3 customized models to triangulate the best target drugs. The training data for the GlimmerX machine learning system is the GlimDS dataset. That is 400 of the newly assembled protein-drug pairs as inputs, labeled with the interaction score of the drug and protein, as well as controls so that the model would learn what it looks like to not interact with the glymphatic system. The GlimmerX machine learning system has 3 different models that I created, one based on a KNN architecture, one based on an XGBoost architecture, and one a neural network. In my final implementation stage, I ran 11,575 FDA-approved drugs to be evaluated by all 3 models, and if a drug was determined to have a high interaction score by all 3, it was identified as a drug candidate. The first step in the GlimmerX machine learning system creation was preprocessing. Preprocessing happens to avoid overfitting, which is when the machine learning model becomes too specified to the training data and isn't able to extrapolate its findings to real-world data, obviously something to avoid. Preprocessing narrows the amount of data that the model must interpret, which improves accuracy. I did this using the RDKit molecular fingerprint extraction tool on the drug structure molecules, which extracts the most valuable portions of the molecule and converts it into machine-readable binary. Each of the 3 models included in the GlimmerX machine learning model had their architecture chosen because of a quality that I wanted the final system to have. GlimmerX's first model uses KNN architecture because KNNs easily learn nonlinear decision boundaries. A KNN model is a supervised learning regression algorithm that estimates the proximity of the relations between test cases to the relations between training data. You can see a simplified example of this in Figure 7, where in this graph it's showing the classification of a new data point into category A or B based on which one it's closer to on a graph. A more complicated version of this was implemented to determine whether the drugs were closer to drugs that interacted with proteins in the glymphatic system or not, and a number of hyperparameters were adjusted to improve performance of the KNN. GlimmerX's second model used an XGBoost architecture because it's designed to handle large data sets efficiently. XGBoost architectures are comprised of a series of decision trees that implement parallel tree boosting, which is combining many decision trees together to boost the accuracy of each. You can see in Figure 8 a diagram of how an XGBoost architecture is structured with a number of simple decision trees, but each output of the decision trees affecting the input of the next, forming a complex XGBoost architecture. Again, a number of hyperparameters were adjusted to improve performance of the XGBoost model. GlimmerX's third model is a deep neural network architecture chosen because it can discover complex relationships. Neural networks are comprised of a series of layers like in Figure 9, with an input layer, multiple hidden layers, and an output layer. Each layer is made up of nodes that perform calculations that weight different attributes or features of the drug based on their level of importance in drug-protein interaction detection. Weights are then fed through the activation function, which produces the weights for the next layer as the output, and then the final output is a customized algorithm designed by the machine learning model to predict drug-protein interactions. GlimmerX's deep neural network has 8 hidden layers, which are 5 densely connected layers that perform the weight customization, 2 Gaussian noise layers, which are regularization layers that add random noise to avoid overfitting, and 1 flattened layer, which makes the output of the machine learning model one-dimensional so it can be a single interaction score. Again, in Table 1, you can see the hyperparameters that I adjusted for the GlimmerX deep neural network. All 3 GlimmerX models showed high levels of accuracy. Accuracy was measured with multiple metrics because since this is a regression task, not classification, it's predicting on a continuous scale instead of discrete, so there's no single value for how well it performed on the training data. I used test accuracy, which is how well the model performed on a segregated portion of the training data that the model wasn't trained on, and then was fed back into the model to test how it would perform in a real-world environment. I also used root mean squared error and mean absolute error to see how the model was performing as it was training to make sure that these loss values were going down, an example of which you can see in the graph on this slide, the graph of the neural network loss decreasing over training time. Now for implementation.
Minutes.
11,575 drugs were tested by all three machine learning models, and 273 of them were predicted to have high interactions with glymphatic-related proteins. It's important to note that since all three models were being used, the total Glimerix machine learning system has a higher accuracy than any of the individual models, since each model can catch the errors of each other in a checks and balances type system. Now, future research can target to the 2% of drugs most likely to interact with the glymphatic system. Glimerix using GlimDS can aid researchers in drug discovery by concentrating resources on drugs most likely to have interactions. We can compare this to the current timeline seen in Figure 11, where it normally takes 6 years to narrow down 10,000 compounds to 250, whereas the novel Glimerix machine learning system can narrow down a comparable amount in minutes. This 6 years is 6 years of time, of money, of resources, and most importantly, time the people who need these cures are not getting them. Thus, the Glimerix machine learning system can have a meaningful impact in the fight against neurological disorders. I'd like to thank my science research teacher, Mr. Andrew Bramante, for his amazing support throughout this project, Dr. Zoltan Desso for his generous help and advice on data preprocessing for machine learning, and a huge thank you to the organizers of JSHS for their hard work in making this opportunity possible for all of us. Thank you. Should I keep sharing my screen?